All right, guys, so we're continuing with Unit 7 on adaptations and evolution. Last class, we talked of just kind of like an overview of what it means to be a species and how we came about with the different groups of taxonomy and things like that. Using the work of Carolus Linnaeus, we kind of talked a little bit about Charles Darwin and him being the father of evolution and believing that evolution comes about through natural selection. We talked about the fact that natural selection can only reshuffle the genes that are already available in a population, and environmental pressures actually drive natural selection. So, based on the environment, organisms that are better suited to survive are going to be the ones that are going to have greater reproductive success. We also talked about the fact that mutations are the only way to create new genes, that natural selection does not create new genes, it can only reshuffle the genes. And the key to all of it is that individuals do not evolve, populations evolve. And we know that a population is a group of the same species living in the same area at the same time. We also kind of looked ahead at what are some things that actually cause reproductive isolations. And we looked at the pre-zygotic and the post-zygotic isolations, and these things are things that lead to speciation. We talked about the fact that a species is an organ or two organisms that can produce fertile offspring. So they have to be able to make babies that can make babies. That's what classifies two organisms as being of the same species. If they are unable to do that for any reason, then we consider the organisms to be not the same species. And again, I'm saying organisms, but keep in mind I'm talking about populations, because the inability of one organism within a species to not reproduce does not make them not a part of that species, okay? So I'm talking about, like, in general, members of one population being able to produce fertile offspring makes them of the same species. All right, so let's look at some evidence. What are some things? So we know evolution is just change over time, right? It's the accumulated changes over time that can lead to phenotypic or genotypic or phenotypic differences. Evolution is supported by an overwhelming amount of information. The four key things that support evolution are direct observation, homology, the fossil record, and biogeography. So when we think about direct observation, when we talk about that, there is evidence that we can see in insects and bacteria. We can actually see it directly where they become like insects that are exposed to chemicals like DDT, which is an insecticide. There are some that were able to survive the insecticide and then they reproduce, and then you have insects that become resistant to that particular insecticide. And so now you have what was an original population which was acceptable because a few individuals were able to survive. They passed on that favorable trait for that environmental pressure, and now they have essentially evolved or changed over time so that insecticide no longer works against that particular population of insects. We see it in bacteria all the time. That's why we have MRSA and VRSA, which are methicillin resistant Staphylococcus aureus and vancomycin resistant Staphylococcus aureus. It comes from the overuse of antibiotics and the incorrect use of antibiotics. That's one reason why, if you ever get an antibiotic, what do they tell you to do? Take it all. They tell you to take it all. And the reason they're telling you to take it all is because anytime you don't, you are increasing the chance that some bacteria will survive, and the ones that would have been susceptible can undergo transformation. Remember we talked about transformation. Remember the rat experiment with the heat-killed pathogenic bacteria and the non-pathogenic bacteria? The non-pathogenic bacteria was able to pick up the gene that made them pathogenic. So you run the risk of doing that if you don't take your full antibiotic prescription. And of course, we have an overuse of antibiotics. So we can directly observe. We have been able to directly observe evolution in action, especially in insects and prokaryotes. Viruses also evolve to become resistant. Like, they're not considered living things, but they evolve. That's one of the reasons why some vaccines work 1 year and the next year they don't. All right, homology. What does homo mean? All right, so homology means that you can look at organisms and you can see some similarities between these different organisms. One thing we can look at is called embryology, which is the embryos. So we can look at the actual embryology. And I don't know if you guys remember, we were watching that video of the cell developing. I said you used to look like that at one point. Like, you can look at embryos, and a lot of embryos look exactly the same at the beginning of development, and then they begin to change more and more as development occurs. But embryology is one thing that we can look at, where we look at organisms and it's like they started off as one cell, the one cell became two, two became four, four became eight, and so on. And then the early embryos actually look alike, where they have the tail and they have all of these different similarities, but then over time they begin to differentiate into what the different organism actually looks like. I'll show you a picture of that. Should I already had this up here? Because I knew it was going to move slow. It always does. That one. Okay. So here you see a shark, lizard, chicken, pig, and human. And these are the early embryos. What do you notice about some of these embryos? They look very similar, right, at the beginning. So this is what we mean by embryology, where the beginning stages, even though these would be totally different organisms, right? They are in different kingdoms, but they're in the same kingdom. They're in different classes and orders and families and genus and species. But in the beginning, they pretty much look about the same. Okay, so this is what we mean by embryology. The other thing we have are called vestigial organs. Do y'all know what vestigial organs are? A vestigial organ. I didn't even read I had most on that. Or organs that are no longer needed anymore. Okay? It's like they're there, they exist, but they serve no purpose. For instance, whales have femurs. Did you know that?
No.
Whales have femurs. Do they have legs? Do they need the femurs? No. So that's a vestigial organ. Your wisdom teeth are considered a vestigial organ. Some people consider the appendix a vestigial organ. Although there is some connection to the way your immune system works with the appendix, but you can live just fine without it. What about the what, sweetie?
I was gonna ask the gallbladder because I just heard many people without that surgery.
It is not vestigial because once you live without it? Yes. But do you need it? Yes. Do you live like it? I had my gallbladder removed not this January but last January because I had gallstones, and you have to greatly alter your diet. Because what your gallbladder does is it holds bile, and bile is released in response to breaking down fat. So you need that to break down fat, because anything your body can't break down, it gets rid of. So if you don't have a gallbladder and you eat fatty foods, guess what happens? Guess where you're taking the trip to real soon? Yep. It's either coming up or it's coming.
Yeah.
There you go.
So how are gallstones caused?
It's like a calcification, like you get like, it's kind of hard to explain. Like, you really get it from eating a diet that is very high in fat, because you're basically overworking your gallbladder, and they block the duct that allow the bile to actually escape from the gallbladder and get, the gallstone does. And so that's what causes the pain. Like, so you'll eat something fatty, and then the gallbladder is trying to do its job, but gallstones are blocking it. So it literally feels like someone is taking a hot knife and like sticking it like right here and just turning it over and over, and there's nothing you can do to get rid of that pain. Worst feeling in the world. It literally feels like a mini heart attack. But it is very common for people in southern states to end up having their gallbladder removed when they get older, because of our fatty diets. We eat a lot of fat. It's kind of hard to explain exactly how they form, but if you ever have a problem with it, you're gonna know it. You ever have to get it removed, You would definitely remember when you eat fried chicken. Anyway, certain things you don't want to eat out at restaurants anymore. Because it happens quick. All right, anyway, moving on. But no, it's not a vestigial organ because it does serve a purpose. But vestigial organs are those things that you no longer, that are still in existence, but they're not actually being used, okay? We also see homologous structures. That was the other one I wanted to pull up. Let me go back here. Homologous structures. Da, 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 da. All right, homologous structures are structures that, for instance, okay, I had the wrong thing. That you have a similar actual structure, so it develops from the similar tissues and it has a similar arrangement. So that homologous structures are an indicator of a common ancestor, okay? So here we have a human arm, a dog's leg, a bird's wing, and a whale's fin. And the actual structure, the bones, it has the same types of bones. They're just arranged differently to suit the purpose. But the fact that you have the same types of bones and a similar arrangement is an indicator, it's taken as evidence of a common ancestor between these organisms. Now there's a difference between homologous structures and analogous structures, okay? So homologous structures are an indicator of a common ancestor. Analogous structures, however, are an indicator of common environmental pressures. Does not mean you had a common ancestor. So if I look at a bird's wing and a human's arm, I'm going to see a similar arrangement of the same types of bones. That is considered homologous structures. But if I look at a bird's wing and a butterfly's wing, do they have a similar arrangement of structures? No, because butterflies don't have bones. They're invertebrates, right? But do they have a similar purpose? Yes. That's analogous, okay? Analogous does not indicate a common ancestor. It indicates a common usage. So an analogous structures are two structures where they have the same purpose, but they're made differently. So a bird has a wing for flight. A butterfly has a wing for flight. Birds have bones in their wings. Butterflies don't, but they can both fly. But if you were to look at a human's arm and a bird's wing, the structure and types of bones are similar, but they have two different purposes. So homologous, common structure, different purpose. Analogous, common purpose, different structure. Got it? Homology indicates common ancestor. We also can see molecular homologies on the molecular level where the amino acid sequences are similar, the DNA sequences are similar. So we have, that's called a molecular homology. So these can be molecular as well. All right. The other one is the fossil record. So this is number 2. 2 was homology. Remember, 1 was direct observation. So we had direct observation. 2 was homology. 3, these are evidences for evolution. The fossil record. We can actually examine fossils. And we can look at fossils that have been dated to be very old, and we can take that fossil record all the way up to the present, and we can look at how things have changed or stayed the same, because not everything undergoes evolution at a rapid rate. So the fossil record, all right, we have transitional fossils that have been found that actually link ancient fossils to current organisms. So we just look at what's left behind. A problem with the fossil record, though, is that it is disproportionately available resources for vertebrates versus invertebrates. Why do you think that's true?
Because the invertebrates don't have bones.
They didn't have bone, right? So it really wasn't much to fossilize. Like you may get it like you may get a leaf implant or what's it called, a trilobite, and I always mispronounce that word. Trilobite, that's the one. You may get something like that, but it's very rare to find fossils from plants and invertebrates. Most of the fossils we find are going to be from vertebrates. So we know a lot about vertebrate fossils, but not very much about invertebrate fossils. And the last one, the fourth one, is biogeography. The continents as we know them now were believed to once be one piece of land called what?
Pangea.
Pangea. What happened? What is believed to have happened in Pangea? The tectonic plates begin to do what? Shift. And what happened to Pangea? It separated it. So this is called continental drift, okay? It's when the continents drifted away from each other. And evidence of that would be found in species that can't cross like from one continent to the other. So you have on one continent, you'll have species that are very similar to some on a different continent, which indicates that they may have once been a population that was split due to continental drift. You also have what's called endemic species, which are things that are found in one area but nowhere else. What location is known for its endemic species? Australia. It's so much stuff in Australia that you would never see anywhere else, okay? So all of that is used as evidence of evolution. These are considered the evidence of evolution. Any questions about this? At what level can evolution occur? At what level, what ecological level does evolution occur? Do y'all know what I mean when I say ecological levels?
The kingdom domain.
Well, no, that's nomenclature, but ecological levels would be like once you get down to this species, which is also called an organism. So you have organism or species, right? So when you get a group of these together, they create what's called a population. I can't spell today. I can't spell most days. And then if you consider all the populations in that area, we call that a community. Community is composed of only the biotic or living things in the area. If you decide to include the non-living with the living, then we call this an ecosystem. If the ecosystem has similar weather, it's a large area and has similar weather patterns and climate, then we call that a biome. And then when we group all of the world's biomes together, we call that the biosphere, which is another word for the earth. All right. So when I say ecological levels, this is the level I'm talking about. So from species, when you group species together, we create a population. If we take different populations, like a population of people, a population of pigs, a population of trees, a population of grass, and we count all of that in a habitat, the area, then that becomes a community. And then if we include the ponds and the energy sources, the rocks, the soil, the air content, then we call that the ecosystem, okay? So at which ecological level does evolution occur? Population. Because organisms do not evolve, populations do, okay? Populations evolve. That's important that you remember that. Organisms do not evolve, populations do. So populations evolve due to genetic variation. Genetic variation makes evolution possible. What's the only way to get genetic variation? Mutations. They're the only source of new genes or alleles. We talked about mutations earlier. We talked about point mutations, which are mutations in a gene. We talked about chromosomal mutations, where you can have things like a change in structure or a change in number of chromosomes, okay? Not all mutations are harmful. Normally when we think of mutations, we think that it's harmful, creates a disease, is bad. But not all mutations are harmful. The reason why we have different eye colors is because of a mutation. Was it harmful? No. The reason why we have different hair colors is because of a mutation. Is that harmful? No. Artificial maybe, all them chemicals seeping into your skin, but hey. There's three mechanisms for reshuffling alleles. What are alleles? What are alleles? What are alleles? Can't forget what you've already learned. Alleles.
Can't think of the definition.
The thing with like the planets, I just don't know.
Wow. Alleles are different versions of a gene. It's a gene, but it's different versions of it. That's all alleles are. Different versions of a gene. This is why we have genetic diversity, because there's more than one version for the gene. There's an allele for blue eyes, an allele for green eyes, an allele for brown eyes. And because of that, we have genetic diversity. And so you can reshuffle those alleles three ways. And those three ways include crossing over. When does crossing over happen? Not metaphase. Prophase 1 of meiosis. Crossing over happens during prophase 1 of meiosis. So one way we reshuffle alleles is through crossing over. The other way is through independent assortment, which is when the alleles separate from each other independently. There's an equal chance they can end up in a gamete. You don't know, you can't control which gamete gets which alleles. And the last way is through random fertilization. You don't control the genes that are in the sperm that fertilize the egg naturally. Like, we manipulate stuff as humans, but naturally you can't do that. All right, so we can test whether or not a population is evolving using my favorite thing in this unit, Hardy-Weinberg equilibrium. All right, open your computers and go to the home page, and there is a worksheet there. It's under, if you go to worksheets, it's going to say Hardy-Weinberg practice problems 1 through 5. Just open that up, and we're going to talk about what Hardy-Weinberg is, okay?
Where is it again?
From the home page, where it says worksheets, you have those pictures there that list the different things.
Handouts.
Sean, do you have a utensil for eating?
Yeah, I do not. Alright, so Hardy-Weinberg equilibrium is a test that you use to tell whether or not a population is evolving. Alright? Now, we talked about last time how you have a gene pool, right? And so a gene pool is consistent of all of the different alleles that are available. Like in this room, we have a gene pool. We don't all have the exact same alleles, right? Hello, heard to my people. All right, so in order for the Hardy-Weinberg equilibrium to actually work, there's 5 conditions that must be in place. I need you to hold your fingers up because we're going to use your hands to help you remember. All right, first thing is, let's start with let's see, which one of these we want to do? Let's start with the thumb. All right, is this your biggest finger or your smallest finger? Well, is it a finger?
Yeah.
No.
No.
It's not even a finger, right? So we got fingers and we got a thing that's not a finger. So, is that bigger or smaller than these? Come on now. Who says bigger?
It's fatter.
It's fatter. It's thicker. All right, so I was going for bigger because it is bigger. So you want a large population. So think of your thumb as being bigger, and then do this and say large population. All right, point. I'm telling you to go somewhere. If you think about that in ecology, what is that? If you go with immigration. So you immigration or immigration, okay? So large population, immigration or immigration. Do it. All right. Now, this next finger, would you want to put that one up by itself?
No.
Don't naturally select that one, okay? So no natural selection. Block it off. No natural selection. Do it. No natural selection. All right. What's this finger used for? A sign of what?
Marriage.
Marriage. And so the point of marriage is to mate. So you must have random mating. Got it? All right. And the last one is no changes in the allele frequency. Okay? This one don't really matter, but hey, I mean, can't really come up with no doubt. So what's this one?
No natural selection. Mating.
Random mating and
No change in alleles.
no change in alleles. Do it again.
Population. Immigration. No natural selection.
Random mating.
No change in alleles.
Those are the 5 factors that must be in place for Hardy-Weinberg to work. Now, you've probably seen this equation somewhere before in math, but we're going to use the letters P and Q. Instead, you probably saw it as A squared plus 2AB plus B squared equals 1. What is that? Pythagorean theory, right? Is it Pythagorean? What is, no, it's not Pythagorean. What is it called? I don't know. I don't teach math. What is it?
Wait.
Is it the quadratic?
Tell me to look it up.
Yeah. Oh, okay. Y'all look it up real quick.
This is just factoring. A factored
Well,
out
I know like they
equation.
do that in like, yeah.
You are amazing.
But this is a
Bro,
non-factored
where's
out
this
equation.
thing?
Sorry.
Well, they do that in like the, what is it called?
Oh, yes. Glad we weren't playing from this.
Do you get any scholarship out of this?
I would have been hard
You don't get scholarship money for any award that awards that.
I need to get this out. Oh wait, it's still videoing. Should I give you?
Hello?
No.
I want to, like really badly.
Oh, you eating your thingies?
Yes, I am.
Excuse me, I'm just like, sorry. You're good, you're good. I'm trying to do it quick.
That was unnecessary.
I know, I just wanted to be in it. Hopefully she doesn't want to.
She's posting it. That's why she's.
I know. I don't want her to watch
Hey,
it.
edit this part out, Miss Young.
Don't edit it out.
She's not gonna edit it out.
Oh, this is all soggy, man. Oh, this smells so good.
My computer's gonna die. Ryan, how how much is
Do you
your
like
computer
meatball
charge?
subs, Ryan?
My computer's on 33. I think she needs to.
Alright, so, which one is it? What is it called, y'all? What's this? Y'all done seen it before. I don't do math.
Factoring.
Yeah.
It's what?
Before factoring out.
Okay, whatever works. Alright, so in this case, okay, what's this again?
Population. Immigration. No natural selection. Random mating. No.
Alright, normally I do this, but, you know, cover it up, you say. No natural selection. Alright, anyway. So, Q represents the recessive allele in a population. Alright, so this only works when we have complete dominance, okay? Only works when we have complete dominance, and Q represents the recessive allele. Got it? You with me? Alright, now, you must have the value of Q or Q squared first in order to work this equation. You must solve for Q first. The reason being, if I have a phenotype that is recessive, I automatically know what the alleles are. But if I have a dominant phenotype, it can be homozygous or heterozygous. So The letter Q represents the recessive allele, and the letter P represents the dominant allele. Got it? Now, that means that Q squared means I am homozygous recessive. P squared means I'm homozygous dominant. And PQ means I am heterozygous. Y'all got it? So and so why do you think there's two PQs? If you take two parents who are completely heterozygous, what's your ratio? 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. The total number of dominant and recessive alleles in a population comes up to 100% of the alleles in the gene pool. That's why P plus Q equals 1. Are you following? So when you use Hardy-Weinberg, you need to solve for Q first, and then after you find Q, you can find P, and then you can plug in. So let's kind of, let's just do it, so that way it makes more sense when you actually do it. All right, so on the practice problems, on the first one, I can't see it. Can y'all see it? I printed it out and I still can't see it.
I couldn't, wait, where'd you say it was?
From the home page, you click on the tab that says handouts, and then there's one that says Hardy-Weinberg practice problems, set 1.
You asked me about first question.
There's so many.
The first one was like numbers.
The first question says, what is the Hardy-Weinberg symbol for the frequency of the dominant allele?
Yeah.
The frequency of the dominant allele is what?
P.
P. Well, okay, what's the frequency of the recessive allele?
Q.
Q. What's the symbol for the frequency of homozygous dominant genotype?
Squared.
What squared?
P or Q.
No.
P.
P squared is dominant. What's Q squared? Recessive. Homozygous recessive. So the frequency of the homozygous dominant is P squared, and the frequency of the heterozygous is going to be what?
Q squared. No, 2PQ.
2PQ. All right? 2PQ. Frequency of homozygous recessive is
Q squared.
Q squared. So let's do this one. It says if allele frequency of R is 0.75, what is the allele frequency of big R? So I have little r equals, little r equals 0.75, and I'm looking for big R. All right? So what letter does little r represent in our equation?
Q.
Q. And what's the big R?
P.
P. And what's the equation we're going to use this time?
Q plus P.
P plus Q equals 1. So we know Q. So we have X plus 0.75 equals 1. What is X? 0.25. So what's the frequency of dominant alleles in the population? Which is what percent? 25%. Okay? Simple enough, right? Straightforward. Knowing that recessive is Q and dominant is P, and then using the formula. All right, let's do the next one. It says the allele frequency of R is 0.15. Big R. What's the allele frequency of little r? If big R is 0.15, what's little r? 0.85. So what percentage of the population would have a recessive allele? 85%. What percentage of the population has a dominant allele?
15.
All right, now let's take it up a notch. This is just straightforward when we're using P plus Q. Now, the next one says, in a population, the homozygous dominant individuals make up 81% of the population. So 81% is homozygous dominant. The heterozygote individuals make up 19%. And homozygous recessive make up 1%. What's the frequency of the P allele?
That's
What's the frequency of P?
81.
No.
20 or 21.
No, the frequency of P cannot be 81
Because it's hetero.
So what do we mean?
because 81% were this. So I'm just going to use the letter P, right? 81% was PP. 19% was PQ and 1% was QQ. So you have to use the equation. So what this tells me is 1% is Q squared. And what did I tell you have to solve for first?
I was looking at it.
Q.
P being a lowercase in the thing.
Oh no, P means dominant, Q means recessive. All right, so you have to solve for Q squared. So what's 1% if I change it to a decimal?
0.01.
All right, so that means that Q squared is equal to 0.01. I have to solve for Q. So in order to solve for Q, I take the square root of both sides. So Q is going to be the square root of 0.01. What's the square root of 0.01? Somebody with a calculator, because I don't have one up here. You definitely don't need a calculator for this unit. What's the square root of 0.01?
That's what I figured.
I think it's 0.1, but I'm not sure.
It's 0.1.
0.1. So Q is equal to 0.1. And so if Q is 0.1, then P plus Q equals 1. So X plus 0.1 equals 1. So, well, P, shoot. Y'all know what I was changing it to, I always change it to algebra in my head. So if Q is 0.1, that means P is what? 0.9. P has to be 0.9. So what percentage would that be if you had 0.9? 90%. So the frequency of the P allele in that population is 90%. It gave you P squared, 2PQ, and Q squared. You have to solve for Q first. You solve for Q. It gave you Q squared value, not Q value. So you have to take the square root to find out Q. All right, let's do another one. And y'all wake up, so we can be finished. Tay-Sachs is called by a recessive allele. The frequency of this allele is 0.1 in a population of 3,600. The frequency of this allele is 0.1 in a population of 3,600. What's the frequency of the dominant allele? 0.9. How many people in the population will have Tay-Sachs?
360.
Nope. Tay-Sachs is a recessive, it's a disease caused by a recessive allele. So that means in order to have Tay-Sachs, you must be homozygous recessive. So you must be Q squared to get Tay-Sachs. Understand? The frequency of the recessive allele is 0.01. So Q is equal to 0.1. What's Q squared? What's Q squared? 0.01. So that means Q squared is 0.01. So what percentage is that? This is 1% of the population. What's your population size?
3,600.
3,600. So how many people in a population of that size do you expect to have Tay-Sachs? What's 1% of 3,600? 36. Following? Are we following?
Yes.
How many people in this population of population of Tay-Sachs are carriers?
324.
Walk me through the math.
360 divided or subtracted about 36.
Huh?
There's 10 or .1 that get the recessive level.
Okay, so that means, what's your p-value? 0.9. All right. And how do I figure out heterozygotes? What's the thing for heterozygotes? 2pq. So that means I have to do 2pq is going to give me what I'm looking for. So I multiply 2 times what's p? 0.9. What's q? 0.1 equals what? What's 2 times 0.9 times 0.1?
0.18.
What percent? That's 18%, and 18% of 3,600 is what?
648.
You already did the math? Okay.
It's just,
So that means I expect how many people from the population to be heterozygous? 648. Guys, you gotta follow the equation, you gotta do the math. Gotta follow the equations, gotta do the math. Gotta have a calculator. Alright, let's do 10. Let's do number 10. Number 10 says cystic fibrosis is caused by a recessive allele. The frequency of the allele is 0.1 in a population of 2,500. What's the frequency of the dominant allele? How many people will have cystic fibrosis?
25.
And how many people in the population will be carriers?
450.
And you know that because,
2pq.
and it's literally the same thing as the one above, it just has a different population size. All right, so you know it's 18% of 2,500. Huntington's disease is a dominant allele disorder. While looking at the family's pedigree, Parker noticed that 64 out of 100 of his relatives were affected by the disorder. What's the value of Q squared? All right, so this time we have one that is dominant, right? Huntington's is dominant. So Huntington's is what? What's the values in Hardy-Weinberg? It could be what symbol?
P.
P is a dominant allele, but that's just the allele. This is the phenotype, so what could the phenotypes be?
P squared or PQ.
It could be P squared plus 2PQ. So all of this comes out to be 64 of the total 100. Got it? So how many people were Q squared?
36.
36 were Q squared. All right, so 36 out of 100. So what's the decimal value for this? So that means 0.64 were this. So Q squared was point what? 36. So what's the value of Q squared? 0.36. Okay. What's the value of 2PQ? Tell me how to figure out 2PQ. What's the first thing I need to do?
Can I tell you the answer first? Just to see.
I don't have the answer written down up here, so I wouldn't even.
0.8.
I don't know. Let's work it out.
Wait, no, I know you're talking about.
Let's work it. Let's stop guessing and let's actually work it out. Tell me what's the first thing you need to do. Come on, y'all. You got to make a connection to this.
0.36.
Do what the 0.36? Square root of 0.36. Why are we doing the square root of 0.36?
Q.
To find Q. All right, what's the square root of 0.36?
0.6.
So Q is equal to 0.6. So what does that mean P is equal to? What's P if 0.4? So that means 2PQ is 2 times 0.6 times 0.4. What's 2PQ?
0.48.
0.48. That's the value. So what percentage of the people in the population are heterozygotes? 48%. If the population is 100, how many people were heterozygous? 48, right? 48 people were heterozygous. And so the other were homozygous. Got it?
Yeah.
You sure?
Yeah.
That was C and D. Alright, if you notice, there's, how many sets on here? 3. There's 5 sets of problems. Alright, I'm going to sit, this is where we're going today. Work the second set, and then we'll go over the answers. Work set 2, and we'll go over the answers. Gracie, can you hit that? I forgot I was recording. Can you hit that for me?